the goal of this talk is how do we upscale that information? So what are the challenges? What are the things that we are missing in the current data sets, and how we can transfer that information for modeling workflows. So the title of the talk is Soil Twin, a digital twin for enabling ModEx, that is a model data experiment integration for upscaling these imaging data sets for process models such as PFLOTRAN. So we'll be talking about that in the next couple of slides. So I made a effort to call out the collaborators from EMSL, PNL, and various users who helped to chime in on various of these ideas across US. So this is no means exhaustive. And so let's get started. So why do we need to use this information into the modeling? So as you know, soil is very highly porous depending on the type of soil core you extract. So the main thing is soil pore controls mainly the carbon storage, movement, and transportation, and the pore matters. So as you can see from this figure, that is from a recent Nature Communication paper that is highly cited, in which XCT and SCM EDX datasets are used to understand the pore structure and the chemical information in the pores. So the top figure, the green one, shows the soil matrix, the yellow color information shows the soil pores, and the brown color information is the soil grains. So this is the like for as you move from one aggregate to another aggregate, the soil structure changes. And what are we trying to do is, like, within this varying soil aggregates, how does the chemical information or how does the carbon in this various aggregates spread out? So the lighter purple color represents low carbon content, and the darker ones represent the higher carbon content. As you can see, across the soil aggregates, you can see you can see the carbon concentration being changed dramatically. And what influences that? The pore structure. And in the right hand side of the figure, you can see the pore structure is very important, because in a well-structured soil, you see a lot of pores, that is these white spaces between the soil grains. And in a compacted soil, you see the pores are being very close to each other. There is not that much of connectivity between one pore to another pore. Why that does matter? Mostly it is about how the carbon is being stored within these pores, and how these carbon is being accessible for the crop growth. For example, in a highly porous structure or a well-structured soil, the plant growth is very good compared to a compacted soil, in which the root doesn't have access to the pores and doesn't have access to the nutrients and the carbon for it to degrade or grow. So that's a major challenge. So understanding the pore structure and how the carbon is distributed in the pore structure is very important. And in my title slide, I had the PFLOTRAN. So what does PFLOTRAN do? PFLOTRAN is a modeling, it is a DOE multiphysics code that allows you to develop predictive models for carbon flow and degradation. So what goes into the PFLOTRAN? So you can give experimental data on the soils, such as XCT data, the chemical information into it, and that is being translated into some of the key parameters, such as the permeability, how permeable is the soil, the porosity, and the chemical information, that is the initial condition for how the soil core is being there, associated reaction rates, so how the soil is being, like, how the carbon is being degraded. So once we have that information, we can know how the carbon moves and degrades within the soil. So that's where PFLOTRAN is the DOE code that allows you to ingest this experimental data and predict the carbon movement and degradation. So why PFLOTRAN? So PFLOTRAN is widely used across the US and outside the US too. So it has a wide variety of user database, and more than 3,000 to 4,000 people use it on a daily basis. So it is, as you can see, like, it is being used for understanding the flow and transport, for various other aspects related to thermal degradation, and also the mechanics. And the other aspect is it is a highly scalable code. By scaling, we mean, like, so as you can see, the image data sets are really huge, and you have millions of pixels in the image data set, and you stack it up, it's like billions of pixels. So The PFLOTRAN is a software that allows you to analyze this millions and billions of pixels and in a coherent manner. So what I'm showing in this slide is the scaling. That means you can deploy it on a supercomputing machine that is called ALCF, and you can do a scaling analysis. And in this, we have used approximately 8.3 million mesh cells grid, and you're able to solve a single time step within a couple of microseconds. So that's the advantage of using PFLOTRAN, and it has a really good weak scaling. Okay, so why do we need to construct a digital twin, and where does it play a major role? So through the MONet initiative, as you know, is collecting various soil cores within the US. And from the soil cores, we get the experimental data, that is the XCT, SEM-EDX, or other type of hydraulic experiments. And from that, you generate a lot of chemical imaging data, and which you are using to fuse to construct a comprehensive pore structure and chemical imaging. So once you have that, what we want to understand is, if you flush out the carbon in this, how does the fluid flow happen within this core or within each of these elements? So that provides, like, once you have this pore structure, you can use models such as PFLOTRAN to understand the fluid flow and compare that with experimental methods such as the breakthrough curves that you obtain from the hydraulic experiment. So once you have this framework in place, you know where should you sample next. Say, for example, two of the MONet cores have a similar imaging structure. What you can do is, you don't need to run multiple experiments on the same core, because you know that you're going to get the same answer. So that's the reason why a digital twin will allow you to better understand and sample where you should collect the soil core next. So we'll be concentrating on the first part, that is the multimodal data fusion, and tell you the challenges involved in this multimodal data fusion. So what do you mean by modality? So in the previous talks, as you discussed morely on the imaging, so you have 2D and 3D modalities. By 2D modality, we mean SEM-EDX. So you slice the sample, you get the chemical information, and use that information to transfer that to an XCT. So XCT is a 3D modality. You have a stack of 2D images that line up into a 3D modality. And what you get from, you get complementary information from this. So from SEM-EDX, you get chemical abundance information. That is an initial condition for the flow modeling, that is for PFLOTRAN. And from XCT, you get soil structure, that is a permeability, like the porosity and the associated permeability in an indirect fashion. So that is what this 3D XCT image data set provides. No chemistry information, but a grayscale information, providing information on the pore network structure. The SEM-EDX provides a detailed chemical information. So we combine this information to get a comprehensive 3D structure, and we calibrate and validate it through a modality called breakthrough curve. So you perform a hydraulic experiment on the soil core, and you get a time series of how the carbon is being produced over time. And that is used to calibrate your permeability and validate it and see how the XCT complements the pore structure and the permeability within the system. So you combine these three modalities to better understand how the soil behaves and degrades over time in a well-structured and non-well-structured system. So, okay, what is a challenge? Before you use any machine learning methodology, we need to have a really good data set. And having a really good data set is a major challenge, because so from an experimental process, I have seen these images in the morning. So visually, they are really good. So what are the challenges when we are trying to curate this data set? So XCT is a stack of more than a simple aggregate, a first set of aggregate. You have more than 1,700 images, and each of these images is more than 1 million pixels. So a stack itself is more than 1 billion pixels. And that, if you want to resolve it at that scale, that's almost 2 billion mesh cells for PFLOTRAN. And you need to do this for hundreds and thousands of these aggregates as you're collecting more and more experimental data. And the second part is, you are getting these slices from the SEM-EDX, and each of these slices is of a different dimension. So as you can see, even within the elemental dimensions for silica, aluminum, and iron, along with carbon and oxygen, you have different dimensions that you need to curate. And that is very important, because you can't just give this to a machine learning toolbox or a deep neural network toolbox to get the to link whatever is in the XCT to SEM-EDX. So most of the time, it's 80% of curation of the data set for the machine learning, such that once you have that, then you can use various machine learning tools to have a one-to-one mapping between the grayscale and the color scale from the SEM-EDX. Okay, so that is a major challenge. So there is no workflow for EMSL users to fuse this data set and transfer that information to process models such as PFLOTRAN. That's the first thing we want to address. And how do we do that? So the exciting part is like we have this, like the what I'm showing is per one aggregate. We are going to get thousands of these images from various cores and aggregates. And the gap is to accurately map this information to from the grayscale to the chemical scale. As you have seen in the previous talks from Peter and from Sam, so the major challenge is a co-registration. The co-registration means like these two samples have a different resolution, they are deformed, and there's a lot of space that is like there's a lot of background noise that is not important. So we need to overcome the sample deformations, resolution differences between XCT and SEM-EDX. For example, you have a sample that is being deformed like this. You want to map them into one-to-one correspondence between these two and getting the correct XCT slice. So Odeta mentioned in the morning that for one SEM-EDX, there's a stack of images from the XCT that may have a similar correspondence. So we need to identify getting the correct XCT slice, map them up, resolve the differences, and overcome the sample deformation. So these are various big challenges. So how are we gonna, so let me go to the nutshell. So how are we gonna overcome that? So in the SEM-EDX, you have a background. So what we do is we manually mask and label the background with respect to the aggregate. So this is the background that is on my pointer, and this is the aggregate. So first, you use a simple k-means clustering to differentiate between the background and the aggregate. Once you create a mask, you remove the background and extract the information within the aggregate, but you still have a lot of background here. So and it changes from aggregate to aggregate. The second challenge is, okay, the second challenge, let me go into this. So the second challenge is a co-registration. So the raw XCT slice is of one dimension. After you transform and curate, you get a image that is potentially be co-registered with the SEM-EDX. So the image, as you have seen, is of different resolution with respect to SEM-EDX. So what we are looking for as a first step through a vision-based algorithm is for templates. So what are the denser particles and how we map to this? So initial co-registration process is looking for templates that you are able to match between between this image and the other image. So what you are looking for these anchor points, and once you get these anchor points, you are able to create subvolumes. That means you only have 5 slices. So you can't just use a traditional machine learning methodology to use these 5 sizes, because you are going to get an overfitting and you're not going to predict in a right manner. So what we are going to do is we are going to take this image and chunk it into small pieces and curate them in such a way that it is amenable for the machine learning methodology. And what we are going to do is we find the anchor point that is this point here from the traditional template matching, and take that sub volume. So that is of typical dimension of 100 by 100. The reason why we are looking for 100 by 100 is because we want to upscale it. So obviously in the co-registration process, you are going to get errors. So are the errors important? How important are they? So we are going to use a representative volume element to match what is the maximum volume size. So when you're trying to co-register, you're going to get a different image for the SEM-EDX. So we are going to transform that into, we are going to coarsen that into the same size of the the XCT. And we are going to use a part of this image for training the machine learning algorithm. So we are going to use a template in the top 20 by 20 as a training, and remaining for the testing and validation. So once we do that, random, like the reason why we are using random forest is like, it's easier to train. You can train it in like, for me, took no more than a minute to train a sample. So the predictions are reasonably good. So go for the lowest hanging fruit. That is like, this is the baseline model. Anything greater than this is very good. So you can train, test, quickly understand what's going on, and get a reasonably good estimate for the model. But as Odeta showed in the morning, when we transfer this image for the entire SEM-EDX slice, it did not perform well. So the next step is to take these multiple of these sub images and curate a very well organized database so that you can train the random forest on various parts of the subimages and get a comprehensive overview for the SEM EDX. Okay, so what we are going to do, how are we going to upscale? So let me get to the bottom of this quickly. So you have a raw XCT image. We are going to use a segmentation, a simple segmentation, to transform this into a binary image with pore and solid structure. So they should be interlaid. And once we get that information, we are going to use a trained machine learning model on this segmented image to fill in what is a pore and what is a solid. So the darker regions represent the pore and the lighter regions represent the solid. Once we get that information, you mesh it up and use that as an information for the PFLOTRAN modeling. So this is the main part. So once we mesh it up and create a 3D cube of various pixels, we can use the PFLOTRAN capabilities to better model the flow and transport and how the pore carbon or elemental information is being flushed out. We can use those process models to create a really good comprehensive picture of how the flow happens. So our next steps is to better understand and use these process models to see how the carbon degrades, the omics information that you get from other complementary data sets such as FTICR mass specs or other type of metabolomic models, and use that as a basis to construct a comprehensive model for understanding the chemistry in time. So with that, I'm going to conclude. So there was no workflow such as this. So we're trying to make this open source so that people can try and test it and provide feedback. The goal is to construct a comprehensive database so that you can expand this 2D chemical information to 3D space, and potentially more than 12 users are currently testing this workflow and using parts of this workflow in their research. So with that, thank you.